This is Regenerative Skills, the podcast helping you to learn the skills and solutions to create an abundant and connected future. I'm your host, Oliver Gaucher. Are you left wanting more at the end of each episode of this show? Are these short sessions getting you fired up to try new skills for yourself and share the journey with others who are working through the same challenges? Well, the good news is that this podcast is only the beginning. The real action and learning is happening on the Regenerative Skills Discord channel, where you can connect with the whole community to dive deeper into the topics on the show, explore solutions, and share your journey and blooper reel with an active group that can't wait to hear from you. You can get your questions answered and share knowledge and wisdom of your own on a safe platform that, unlike the social media giants, won't steal your personal data to advertise to you in creepy ways. Ditch Facebook and join us where the real skill builders are. Just find the link to the Discord chat on the homepage at regenerativeskills.com. Hey there, everybody, and welcome to the very first episode of Season 9. This is the beginning of the 9th year of making this show. Now, I've got a lot of exciting new projects and collaborations coming up that I'll be bringing you along for, including some ambitious water restoration projects, some educational trainings for regen ag transition in Europe, and quite a few mini-series on new topics and perspectives on regeneration that I'll be co-creating with some friends and members of this podcast Discord server. But before I get into all of that, I want to start this season off with an exploration of the cutting edge of soil science and agronomy, a little preview of what's to come. And as part of the monthly panel discussion series, that's exactly what we focused on in a live interview with Harriet Mela and James White. Now, since they've both been on the show in the past, and they give short intros to themselves at the beginning of the chat, I'll leave it there and we can jump right into the conversation. So, I'm going to do a quick intro to get us kicked off here. So, I'm Oliver Gaucher. I work with climate farmers on the academy side, and this is a production of the nonprofit wing of this organization, where we are trying to bring a community of farmers all over Europe together in order to share knowledge and have access to what's going on in the field and the cutting edge information that's available to help them succeed in their own endeavors. And that's what brings us here today with Harriet and James. We're going to be exploring the cutting edge of soil science and agronomy from some of the people who are at the forefront of this type of research. And, well, I'll let you each introduce yourselves real quickly. So, Harriet, would you like to give us your intro to get us started?
I'm here. Okay, I am a biologist. I studied in Tübingen ages ago, and I work with soils. I work with seeds. I work with people. I like education, and I do product development. And I think that should do.
Perfect. We'll get into the stuff that you're excited about and researching at the moment in just a minute. James, can you give us a short introduction on yourself and your work?
Yes, but I'll add one more thing about Harriet. She has a command of plant physiology like nobody I know. You know, so she knows she's being somewhat modest. Sorry, Harriet. Sorry about that. I'm James White. I'm a professor at Rutgers University in New Jersey. I work on endophytes, and I teach courses here on fungi and plant science and other courses over the years. And I think that's it.
Well, perfect. Let's get started with the first question of our panel today, which is, what information or new discoveries do you find to be most exciting in the areas of soil science and agronomy at the moment, and why? Harriet, would you like to start us off?
Oh, it's me.
I'm going to pick on you all night, so.
Oh my God. Latest discovery in soil science. You know, for me, it's maybe it's this. Maybe it's to shift plant or agriculture around from that view that we're source limited to sink limitation. So we have this true deficiency state when the soils are wrecked and when the plants are in need of minerals and everything, and nutrition is bad and whatever. And we kind of can insert this through several tricks. and shortcut the system and you know. And then comes the moment where the system starts to stagnate and you have this feeling, what's going on? I'm doing my practices, I'm fertilizing, and I'm, you know, and the system doesn't leap, yeah, it doesn't fly. And the understanding that all these things are working in feedback loops, and if the product is not used, that we go into feedback inhibition of production, and that we sit there and have the plants sitting in the furrow and maybe not doing what they should do, not feeding, not producing, because we broke the cycles. So basically, to come to this idea, to understand that a system has to deserve its status itself, and then understand that when the demand is in the whole system and the cycles are closed, that's when it starts to really hum. So, for example, to be concrete, we have photosynthesis. Photosynthesis produces carbohydrates. If these carbohydrates cannot be extracted from the leaves and they accumulate there, they are a problem. Production needs to shut off. If they go and are exported into the root and they are not consumed down there because we have too little oxygen and the microbiome around the root is not consuming that, then these things go back up in the shoot and they switch photosynthesis off. Same thing. And so we have this idea we can maximize photosynthesis and again impose our will on this plant, but in fact we can't. So understanding what we have to do all along this loop so that production and consumption in growth through the microbiome works steadily will just really relieve this system and help the production go steady and production be boosted to another level. And this is this idea to go away from from source limitation to sink limitation, and also go away from this idea of precision control of everything to this idea to understand and be part of a system and get trust in the system to work. And I'm sorry, this is not mainstream soil science because this goes into the direction of control and manipulation. Because every paper you read is like, oh, we found the super tweak now and we're going to make this wow effect and be better than creation intended it. But there is this other trend, which is usually away from these publications, where you see people gain trust and gain a handling of the systems. And this excites me. If that really works, very good.
Fantastic. That's the type of answer that I was counting on from you. James, I'm going to pose the same question to you before we dig deeper into this system understanding of plant and soil health. Can you tell us about what you're most excited about at the moment on the forefront of your own research, or what you're excited to be digging into this upcoming year?
I will. And it's something that we are just beginning to, in science, get an inkling of, and that is that bacteria, for example, soil microbes especially bacteria have a huge effect on the plant, and the possibility is that effect is directly on the genes of the plant, causing additional genetic variability in the plant, and some genetic changes that affect plant expression, the expression of the plant's genome, right? So epigenetics. So that's for the plant, but now talking about soil science, I mean, that's involved there because those microbes are from the soil that the plant is getting. But the other effect is on another group of organisms, and those are the fungal pathogens. And they also will associate intimately with bacteria, including inside their hyphae and vectoring in their spores, for example, when they make spores. And those associations from experiments, we know those associations affect the behavior of the fungus, a fungal pathogen, and at least in many cases, the virulence is reduced when they have bacteria there. There's a few cases where it goes up, but generally it goes down. And so the really what is really new here is beginning to try to sort out how these interactions are happening that favor the plant development, both inside the plant, but also in the soil and the soil environment and potential pathogens in the soil. So understanding and ultimately understanding the genetics of how that happens. So that's what I find. To me, that's the most interesting area right now.
That is fascinating. And I'm actually curious, Harriet, how that fits in with what you were talking about, because James basically outlined some complex relationships and the relationships between different organisms within the soil microbiome. And this seems to me to fit into this larger systemic idea of relationships and cycles, which you were mentioning.
Well, I've spent the last years on some mental hobbies, and one of these is sorting the frame set of interaction of the endophytes with the plant. And what fascinates me really is that you can look in any layer, and for me, it's just the more I know, the more ridiculous I find it to hijack this system with a single compound or something. Because it's like all these safety layers, like for example, plant, when it has to recognize it is a friend or is it an enemy that's just sitting in my roots, or how they control, for example, a mycorrhiza, that it just doesn't grow and drain nutrients, but really work for what it's getting there. And then you see on every organizational level, be it the genes, be it the minerals, be it the chemical communication, be it the structure, it's just interlaced to a degree where you sit there and it's like, wow, it's absolutely fantastic. And then we come along and we think, okay, we just take a plant hormone, like we use auxin and we just pour it on the plant and it becomes bigger. And then it's like, well, yes, more biomass, but has it the minerals or not? And then you find out, okay, you have root microbiome that gets certain amino acids from the plant, and then it transforms them into auxin, and this is taken up from the plant, and then you have these growth effects. And then you think, well, why do they do it? And then you see that the plant, to produce this amino acid, takes zinc. And if it has zinc sufficiency, it will do this, and then you have this whole cycle. And where does the zinc come from? It comes from the microbiome again. And so you see that all this is interlaced on so many levels of interaction, and it must be so. And the big question that I have is, you have all these players there, if you have them. I hope you have them. You have all this diversity of bacteria. You have all these metabolic cycles. You have all these fungi there and everything. And it can immediately go into any direction. All these big circles, to volatilization, to leaching, to feeding the plant, fixation. And the question for me is, why and how does this result in growth? And why and how does this result in destruction? Like James says, you have these bacteria and they go in the hyphae and wow, they're not virulent anymore. But why do they do so? And then we have these super smart people that come along now and say, okay, we just forcefully inject some super bacteria in these fungi, and then we have this super fungus, and then we put this out there and our system is going to make woof. I doubt it. Or manipulate, for example, there is a very new paper about the recognition mechanism. What is a symbiosis? What pathogen? It's like a symbiosis results in very even, rather slow calcium oscillations in the inside of the plant, coming from the nuclear region. And if you have a pathogen, this goes very fast and irregular, or if you have stress, it's just all messed up. And okay, you can go on now and you say, we manipulate this endogenous rhythm then. And then we have a plant that's going to take up the symbionts more readily. Super. And our wheat that is so hard to go along with mycorrhizae, now it will be handshaking much easier, and it will be working with the mycorrhizae. And I look at this and I think, folks, maybe, but maybe not, because it is just interlaced in so many layers, and you have all these metabolic pathways linked to this, and all these signaling mechanisms and everything. You're going to produce another problem on the other end. And I think, why don't we instead invest the energy for this quest on how to get these organisms really settled in the soil and how to let them interact there in a friendly way? This would what I would like to see in terms of research, more, much more.
How to create healthy soils, right? How to create good regenerative soils.
Plant-friendly soils.
Yeah.
Yeah,
And what
how does that fit in with what you were talking about? How does that fit in with the relationship between the pathogenic microbes and the endophytes and, well, the relationships that you outlined earlier? Does that fit in with the model that you're investigating right now?
meaning the model that we're discussing, looking at relationships?
Yeah,
Yeah.
exactly. Instead of manipulating
Yes.
individual parts, looking at it as
Yeah.
this larger ecosystem.
Yeah, I think that's the key, really. I think the ecology of it is, and that's the nice thing about regenerative agriculture, is that growers really have to be ecologists. They have to think like an ecologist. And this is basically what we're talking about. And it's not just, we're talking about, we have to think about plants differently, plants as organisms, plants and fungi that exhibit behaviors, right? You look at their behaviors and you see, with this microbe in the system, then the behavior changes or it doesn't, right? How does that behavior change? So you can look at these interactions that are happening between the microbe and the plant. And some of those endophytes, some of the bacteria that plants take into them, actually go out and also affiliate with fungi in the soil. So even the same organism can create a kind of a bridge between potentially pathogenic fungi in the soil and plants, and alter that association in between between the fungus and the plant. So, and that's fairly common. It's not like an uncommon observation. It's a frequent observation that bacteria will change behavior of fungi. A lot of people, when they look for these kind of things, they'll grow them in culture and they'll just make a little colony of the bacterial colony, then they put the fungus near it, and they look for things that inhibit, right, that make the fungus go away. But that's really not how it works in nature. In nature, mycelium is growing and bacteria will grow right on the surface of them and internally colonize as well. So that's what happens in nature. It doesn't necessarily kill the pathogen. It just alters its behavior, reduces its virulence, or completely eliminates its virulence. And some fungi with bacteria with them, some pathogens, become endophytes because they're no longer virulent. And they actually benefit the plant development. So that whole approach, looking at relationships, yes, I think that's important. And behaviors, that's the other thing. Farmers, growers can, we're really looking at their crops. They're looking at how their crops are behaving, how they're growing and, what's happening out there. So, yeah, I think that they're focusing on relationships, ecological relationships is a key thing.
I really want to go deeper into what you started talking about here, which is how this information, which probably is going over the head of quite a few people, myself included at times, can be translated into practical understanding and action out in the field, right? So with all of these things at the cutting edge of research and this greater understanding of an ecological point of view of what is happening at a microbiological level, How does this translate into action in the field, and what can people do with this understanding when managing their farms and their crops?
Harriet, you want to do that first? You want to go first? You got any ideas?
Mm.
Okay.
Basically, what I see is that we currently have two profitable, actually, possibilities to look at these things. Number one is to create super specific, highly controlled, precise environments and play with these ideas. So you have the highly selected plant that just works with this mycorrhiza, with this bacterial species sitting on this mycorrhiza and in this soil, and with this irrigation and 3 microliters of water on the right leaf. Like, so, this idea. And then, I don't know, high voltage over the head to simulate the thunderstorm in approach like in China. All these things just really. So that's the one thing. And the other thing is to say, I look at it in terms of a local evolution. And I allow the system to self-optimize and to learn. And I entrust myself as part of the system to the idea that it is a self-optimizing system. Because if you look at what Toby Kears, for example, has found, that mycorrhizae, they are trade agents and they go with their materials where they get paid, and they just really shift them from sources where they have high phosphorus to places where there is little phosphorus, and they really bargain for it. They know what a young root is, they know what an old root is, and they go for the host that really pays well. What does that mean? That means that if we have several generations in a certain condition, that the best paying host will have a growth benefit, and this host controls different mycorrhizae around its root, so it selects and feeds the best one most. So this is like a Munchausen system. It's an upward spiral. And that means then when we close these loops again, then when we let it run for several generations, and then we come back to James, we had just recently a discussion after my mycorrhizal talk about the change in amino acid composition, because I wanted to know from him if he was into that. And he said, yes, of course. It's not just the root that enriches and lies in the methionine, it's the shoot as well. And when you look into physiology, you see that methionine, again, it just changes, really literally chemically changes the DNA. It changes the gene activity. So we have this interplay going on. And if we let this run for several generations, so the key is that we really dare to let it happen. And when we
She seems to be freezing right at the most crucial moments. James, can you jump in there for a second?
Yeah, no, I can put this, okay, this concept of the self-optimizing system, soil microbes and endophytes play a big role there. And basically these plants take the microbes out of the soil that they need to survive in that environment. So they essentially adapt, they optimize to that environment, but you have to grow them for a while to do that. So that's a little practical thing there, one of the few practical things that I could think of.
So the concept here is to take more of a hands-off approach if what you're used to is trying to micromanage the balance of nutrients or the presence of certain life forms in the biology. Is that what we're getting at?
Yeah, the point is you build your soil, you build your community so that the plants can actually take care of themselves, so that I mean, yeah, we have one concept, chemical industrial agriculture, where we control everything. We're controlling that environment. We're doing it in a certain way. It's very technician type focused, right? Just everything has to be technically done. But then there's the other way, the regenerative way. Okay. And that is this self-optimizing system that uses microbes and nutrients naturally available in the soil that you're not trying to control everything. Instead, you're trying to optimize plant growth so that the plants can then do what they need to do to grow and they'll adapt. They'll take in microbes that, for example, if it's a bad environment, if it's a bad soil, if it's got too much salt in it or something, they'll take in microbes that help them to grow there. So if you grow the same crop there, especially if you could take your seed and grow them year after year, season after season, then they'll adapt to that soil using the endophytes that are naturally resistant in those environments. So it's exactly that self-optimizing system that involves plants, microbes, and soil, the soil community. And yeah, just following up on Harriet's concept there with something else.
Sure. And I want to play devil's advocate for a minute here, because I can also hear some people thinking, well, I've got crops failing out in the field right now. I don't have time for them to make these connections with endophytes and overcome whatever it is that's causing them, well, to fail, whatever that might be. And what do you say to that? I mean, how do you start to investigate if you don't have this level of understanding of crop health and soil biology to troubleshoot the system to make sure that you can still get a yield at the end of the season?
Well, now you're talking about something truly practical. I can't tell you what to do. I mean, there are situations where the environment is so bad that you're not going to be able to do anything, especially in terms of endophytes, to grow plants in that environment. So if it's a shocking drought, the drought is so deep that plants go, beyond the wilt capacity, no endophyte will help it, right? So you could, in those cases, I don't know what you do. Hopefully crop insurance will save the day, or, not, I couldn't give you advice on that if it doesn't work.
Okay.
Should it be the same question that you asked me, Oliver? The one about something's not working?
How nice is this?
Failing, what do you do?
Yeah, Harriet, we were basically just exploring the practical side of whether the plants and the biology that supports them are able to respond quickly enough to the challenges of, well, the practicalities of working out in the field. If you're at risk of losing a crop, or if you're starting to see animal health decline, do you wait around for the system to self-regulate, to, I guess, bounce back from these missed connections or systems? Or are there ways to intervene, or at least start to diagnose or troubleshoot the system that could give you some insights on how to make corrections?
Look, that depends how cool you are. I recall I talked to Mark Shepard, and I was talking to, and I mean, he's a person that's really cool. He says, if I have a disease, what a bummer.
Yeah, we got you. You were talking about your
This is really silly.
We'll get her back. Until she comes back, I think I could probably fill in what the conversation with Mark was about, because he has the very well-known stun method, where if there is a disease or if there is a challenge in one of his crops, Mark often just lets them die off.
He just lets it run,
and allow them to to bow out, and he just favors the ones that continue to do well over time and that showcase the characteristics of the vigor or the production or the other qualities that he's selecting for, and breed based on the reproduction of those crops or animals as well. And I know that's been a guide for my own farm, and that's kind of how we're approaching this topic as well.
This is kind of create land races, and that's the kind of adaptedness. But it requires really that a grower is able to grow their own seed season after season to do that. And if you're dependent on companies for seed, say it's hybrid seed, you can't do that. You're not supposed to grow those. So there's a certain amount of freedom that you have to have with your own seed, growing your seed, in order to actually build up the microbiomes in plants.
Well, this is kind of what we were talking about the last time we had this three-way conversation with you and Harriet. is that there is a huge amount of untapped potential in going back to community level seed saving and eventually just promoting the genetic material that does well under the conditions and maybe even the deficiencies or the pest pressure that you have in your particular area. Have you played around with that in any of your research?
We're not trying to create land races, but what we do is one of the things that we tend to do is to try to move endophytic microbes from one plant to another. So we'll do things like, for example, take wild weed, a weedy plant that we know is filled with lots of endophytes, based on microscopy examination and other kind of analysis, isolations and so forth. And then we take the microbes out of it, not isolating, but grinding up the plant so that the bacteria come out. And then we put those into, for example, grass seedlings so that we can evaluate what their defects. We're actually trying to figure out how to move microbes from wild plants to cultivated plants so we can essentially restore microbiomes in some crops. So that's the idea. But we do those kind of experiments, moving microbes around between plants to observe how it works. It's very, very similar to the soil thing, where others, for example, there was some work done by Rusty Rodriguez, and I think it was the early 2000s, where they actually looked at some plants, some grass plants growing, panicum growing on a cline from high or low salt to high salt. And they evaluated what endophytes were being taken in as it went through this cline, getting higher and higher salt concentration. They identified a fusarium, I think it was a fusarium species, that they could then take that and put it onto different plants and acquire the same kind of drought tolerance or salt tolerance. And they actually call that phenomenon habitat adaptive symbiosis. It's the idea that plants are getting endophytes that help them to survive in an environment, in specific environments.
Fascinating. I see many parallels to that concept, even transfers of the human microbiome to people who are suffering from gastrointestinal diseases, or, let's face it, making compost or bringing in healthy soil from a forest nearby is a similar concept of kind of inoculating something that has these present cycles and systems intact in them and implanting them in an area where they're deficient to see if you can kickstart that cycle and maybe increase the speed at which that other damaged system can recover. Harriet,
Yeah.
did we have you back already?
I hope so.
Okay, fantastic. All right, well, let's give this another shot. Go for it.
Okay, now I'm going to speak at double speed, to make up for all this fuss. Basically, what we need to understand is that we have several components that need to meet. So there is a lot of talk about endophytes and microbes. There's a lot of talk about mineral nutrition. And what we always forget is that this has to grow into structure. So what we really need to watch and promote is soil structure. All these things can't work properly if we do not have gradients in the soil, crumbs, burrows, and all these things. So the thing is, and for me, it's better to be very imperfect in all of these areas than bring one of these areas into super perfection. So if we have an understanding where we say, for example, I'm this super plant nutritionalist and I know exactly at what phase of the plant I have to apply which nutrient as foliar and how to brew all these, yes, one box. Reduce that to half of the effort, take care of endophytes in the soil, and it's much easier. And the thing is that the more you can delegate to your system and to the system's robustness, the more relaxed you are as a farmer. And this is so hard because you have to be so brave to really slowly wean your system of your intervention. As I said, I'm not cool as Mark Shepard is. In some cases I am. I said, either you grow or you die, I don't care. And then if they did, nice. And if they didn't, okay. But if you really depend on your crop to make an income, it's hard to be so cool. And this is why relevant for farmers, small pilots. And also understand that seasons are not equal, and what works in season X is not going to repeat in season Y. I cannot tell you in these years when I've been gardening how often I thought, now I understood it. This is the way I'm going to do it, always. I've learned to put my eggs in several baskets because, especially in the chaotic climate we had in the last year, it's always good to have several, like seeding directly, preparing some transplants, different dates, and then one of all these things is always going to work. And if 3 fail, who cares? If you have the seeds and they don't cost you much, who cares? And this is back to the seed saving, knowledge about seed saving and seed sharing rings and how the genetic side of it, it's really important, and to do good quality. Because what's very often misunderstood is just you throw a bunch of rascals out there and they're going to do the trick for no, they aren't. You really need to be constantly selecting for what you need, leaving diversity in it, confronting them with new situations, bringing microbiome in. But really, you need to look that what they do is what you want or what you need. So you're a factor in this system. And then the microbiome is, there is so many different classes. And for me, it was appalling to see how much fraud, actually, and I really want to call it like this, is already in bought mycorrhizal inoculants. If 80%, there is meta-studies that have looked at that and say 80% of the mycorrhizal inoculants, they simply don't live up to what they claim. There is no association formed. Some of them have dead spores, some of them have pathogens, whatever. And I mean, then you're as a farmer, you think you're buying the bang in a bottle. It doesn't work. You think mycorrhizae don't work, but in fact, they were just dead or defect, or the system that you have doesn't support them because it doesn't breathe enough. So, or the plant is a dwarf plant and doesn't accept them. And now we become fashionable and we say, okay, we have the mycorrhizae, we have the microalgae, we have this, we have that. And the spectrum is so large. And then farmers very often, they get confused because you think you can't do all of this. So my personal inclination is to do less from this perfection control pathway and go more to black box pathways where you say, I work with soils that I take from one part to the other. And I know and I trust that when a weed has grown in a place and it leaves its microbiome in this place, we know from experiments that always the next plant growing in this place will pick up some endophytes from this place and have them. So this is this succession. And that means, because I have just seen a bit of this question in the chat with the corner of my eye, of course, if we have wild plants, that is weeds, in this system, they bring stuff into it with them. And the question is, and this is the sore spot of all these regenerative ideas, is how do we deal with this? And of course, redefining them from weeds to caretakers of the soil doesn't solve your problem. That if they do their job too well, they just smother your crop. So these management things, how do I deal with weeds? And how do I deal with things that I regard as pests? And how much poison is per default in my system and how much compaction is per default in my system? That's decisive about your success on the microbiome side. And this is something that I find interesting because we talk so long about these things, about weed control and aphids and all these things. And compaction, and it's so boring. I mean, I cannot tell you how long Elaine Ingham talks about aerobic soils and decompaction. And you go out there and you name the field in my vicinity, they're all compacted. No change. So understanding this, and that we need 3 years to establish fully shared metabolic pathways in soils because the division rate is so low. 3 years. You do something today and you will see the full outcome in 3 years. 1,000 generations of microbes. So one way of speeding this up is to say, I incubate them a while together so they have a head start and a bit of a more favorable environment, like soft composting, not hot, colder composting methods. So they can habituate a bit with each other. Yes, it's a selection, we narrow diversity, all of this, but we already have a consortium that knows each other a bit. One possibility. Or else, of course, the foliar feeding hack is always nice. If you see that your system is going down the drain, don't wait too long. The thing is, what I know from experience, when you have set up diversity well and soil is kind of aggregated and you know you have done something for microbiology, in spring, in framing stage, one really loses faith. The plants start to grow and they just don't look good. You know, they grow and they're kind of a bit too yellow for your taste. And you're like, oh my God, and you get really nervous. And they don't grow as fast as when you really give them nitrogen. And you get really nervous. And if you know the system and you see that they are still doing their thing, still catching sunlight, and they're not limp and they're, not really signs of unwell being, but just of nutrient shortage a bit, that's okay. And that's a moment where you can say, I keep cool and I have the trust that this system is going to do it. And usually what you see after a couple of weeks is that the microbes kick in and that they start to really green and go ahead. And when reproduction comes, these plants, they're just really pulled through, while these that you pampered fall off. But this is experience with the system, and I know how nervous you get when you see that. And I know that you really need experience with crop scouting to discern, do I have an illness? Is something going wrong? Do I have adverse microbiome here? Do I need to do something? Should I give a micronutrient foliar? Whatever. Or can I just entrust myself and say, this plant just really is eager? It's like these cow's ears. If they're droopy, this animal has something. And if they lift their leaves and if they attract the sun, you already know this plant is feeding. It's on a good way. And then you can just kind of pimp the system, but don't interfere with it. So this is practical side of it. You need to train your observation. And this is a development, by the way, crop scouting. I just found accidentally crop scouting apps for greenhouses, for peppers and these things. And you have then your camera that runs through there and it just brings back the information, all you need to know. And it gives you better advice and better prognosis of your crop and everything than you could possibly do it. This is how it's marketed. And I'm sure that in many cases this is true because our crop scouting capabilities are really not trained. If you train your observation and you know where to look, you really can extract so much information from being with these plants, the way they smell, the way they move, the way the architecture is, because architecture has a high degree of plasticity. Back to James, epigenetic regulation of the genes through changes in amino acids in the plant, through changes in the mineral nutrition. It's just 100% immediately changes the whole metabolism of the plant and its architecture around and the behavior. And if you can read these signs, you know exactly where your crop stands and you know very well what you can expect from this season. So, of course, you can have the fancy gadget in this controlled environment that tells you the right thing, or else you learn yourself. And sometimes the hard way. I don't want to conceal that because sometimes you really make a mistake. Yes. So learn small.
Man, there's so many amazing nuggets in there that we could spend the rest of the time unpacking. But I do want to bring us back to this last question that I wanted to explore between the two of you, which is, and I guess I'll have to expand it now too. what sort of research or field trials would you like to see happen in the upcoming year in order to advance some of the concepts that you're interested in? But since you had also mentioned about crop scouting and the importance of observation, maybe I would add to that also what sort of educational programs or trainings would also be valuable to see expanded in this upcoming season. Maybe James, you want to take that first?
Yeah, I'll say what the research area I'd like to see. I mean, this whole, from my earlier discussion, this interaction, the genetic interaction, that to me is the new that we really have to understand. Understand first of all the mechanism. How do these microbes, or do these microbes truly have this effect? That's the first question. And it looks like it now based on some circumstantial evidence, but do they really have that effect? And then the question becomes, how do they have that effect? And what is the manifestation of that? Are there more genes? Is methionine, where there's some evidence that that these high microbial corn plants, for example, are loaded with methionine. And so their whole internal genetic architecture changes, and these high microbial corn plants start throwing off mutations. So the other thing is that these bacteria actually colonize the nuclei of plants. So the possibility is that there's some direct interaction between the bacteria and the plant that alters gene expression in the plant. To me, that's the biggest question that I really am most excited about. I don't know if we're going to be able to answer it, but we're trying, and there's some other projects out there that have actually pointed this direction, that there's increased genetic expression in plants with high numbers of endophytic microbes and soil microbes. So to me, that's the biggest area. It's very basic, but I think it's important because it affects, crop adaptation to the environment and a changing climate. You want to be able to enhance evolutionary change in a plant in order to get variations that are going to adapt to the environment. It's like this self-optimizing system, right? I mean, that's part of the changes that plants need to make variation in order to readapt to an environment. Stress, for example, stress requires a lot of changes, and these endophytes, we know that they increase stress tolerance in plants, and part of that may be a direct genetic effect in the plant. So those are areas that I'm interested in, brand new areas that might be important.
Yeah, that's super interesting, especially as we're all battling with a very fast changing climate. The idea that the crops that we plant could make changes or adaptations much more quickly through these relationships is really cool. And like you said, you may not be able to prove something, but that is what makes it exciting, right?
It is.
Harriet, same question to you. What would you like to see more investigation or field trials done on, or perhaps educational trainings?
Learning how to read your plants is certainly one that I think must happen. Otherwise you do not know if you're on the right track or not, especially if you embark on this seed saving avenue. This is one thing. The other thing is, even faster than genetic change of plants, microbe-mediated or not, is okay. If we go back to the very root of these problems that we have, we need aerated soils and a soil sponge. And so the question is, what input channels do we have into these soils? And in general, what you read is photosynthesis, and between the lines is like plants are the only input channel in these soils. And I've spent a while just collecting alternative input channels into soils. And one reason was because in the old biodynamic literature, they have really conducted research where they've shown that bear fallow in winter. So this is the scary ghost of regenerative agriculture. Bear fallow in winter was really giving good yields while building carbon pools in the soil. So this is something where we say, how can that be? And thinking of it, we have, for example, groups like microalgae in the soil and cyanobacteria that even can photosynthesize with far-red infrared light. We have melanin in the soil that can absorb any kind of radiation and kick off electron transfer to microbes. And then we have synthesis facilitated from that input channel. And I'm sure that there is 50 others that I have not thought about, but nature has. So to allow our thinking to expand and go away from this single route to say it must be our crop plants that have to deliver some functions and going and broaden our vision, there is so much metabolic crazy pathways that microbes can do. And the question is rather, how do we guide these things so we have construction instead of destruction in the soil? So we have building of biomass, building of structure, building of consortia. This is something that I would love to see investigated. For example, melanin has this fantastic side effect of being paramagnetic. This is every adept of Callahan is like, ooh, we want to have this. The only way that we can get it is putting basalt in our soils. Nope. Annual lignin apparently can confer this thing also to soils. So all these pathways to get the physical properties into soils are totally neglected. And I think if we would look after this a bit better, and this is really also immediate improvement of the system and this capacity of the soils to deliver growth, I really would enjoy that.
Can I add something?
Yes,
here
please.
That relates to what the microbes are producing, right? What the microbes are doing and delivering to the system and to the plant. But there is another thing that I think needs to be understood, and that is that plants actually can't grow properly without those microbes in their cells, right? So we don't, like, for example, we get no root hairs if you don't have bacteria going in. So this whole question, I mean, it relates to what Harriet's talking about, because some of those nutrients that are some of those things produced by microbes are like hormones, ethylene and auxin and cytokinins. And, you know, it is not fully appreciated that plants actually have to have bacteria inside their cells to grow properly. I mean, I'm not saying they can't divide and produce some cells, but they don't, those cells, those like root hairs don't elongate. You oftentimes lose behavior patterns like gravitropic responsive roots. You get bizarre development if you don't have bacteria there. And then when the bacteria put back on the system, back on the seedlings, they grow quite normally. You know, so we know that there is this, if you look at leaves of plants, you look in the meristems, you see the meristematic cells, for example, tomato, they're full of bacteria in those meristem zones. They're the full extent to which plants actually are a whole community of microbes and the plant is not fully becoming more fully appreciated, but the details and how that works really are not understood. And that's an area that would be really important for people to work on. And it dovetails with Harriet's suggestion.
Yeah, it does indeed. And these are all fascinating things that I would also love to see explored and understood better. And I think that's a good place for us to open it up to questions from the audience. So go ahead and raise your hand digitally there, and I'll call on you in order. And let's make the most of the fact that we've got. All right, so we're leaving it there in the discussion for this podcast version, but if you would like to hear the full Q&A session, you can find the entire recording on the Climate Farmers YouTube channel, and I'll link specifically to this video in the show notes for this episode. So real quickly to end this off, if you're interested in the work that we're doing at the Climate Farmers Academy and would like to sign up, it's free to join for anybody who is actively farming in Europe. You can find the registration link in English, Portuguese, and Spanish at the moment on our website at climatefarmers.org. Now, before we wrap this up, just remember that these episodes are only the beginning of the learning resources, design and coaching services, in-person courses, and interactive community that are available through Regenerative Skills. The Discord server is our free community where you can connect with other like-minded listeners, exchange ideas, stories, tips, and resources, as well as interact with me directly and quite a few former guests from this show. Our Instagram account at regen_skills is the best place to see the projects that me and the team are working on, both for clients and collaborators, as well as on our own properties. I'll also be announcing the certification courses, workshops, and gatherings that we've got coming up later this year. If you're interested in getting dedicated support for your own project, you can now schedule a free planning session with one of our team members through the request form on our website. You can also find all the links, show notes, and past resources there at regenerativeskills.com. We truly believe that no matter your experience, your knowledge, abilities, resources, or background, you can be a powerful force for regeneration on this planet, and we're here to help you find your path. So as always, remember to keep taking those little steps every day towards a regenerative future, and I'll be right by your side along the way.